during animal virus replication, we have a couple of steps that must occur. We have the attachment step. This is right here where our virus is attaching to the outside of the cell. Then we have the penetration step. In penetration, a big difference here is that in animal virus, the entire capsid, the whole virus enters into the cell, becomes part of the cell. There is an additional step here that occurs in penetration in animal viruses that does not occur in bacteriophages, and that is uncoating. Because this virus enters, right? We have our flu virus here, this step A, it's entering into, there's attachment and penetration, and it's in an endosome. Cells take particles in endosomes. But inside of this endosome, somewhere along the way, the capsid protein, that's what this is right here, that capsid protein has to be released. We have to get the genetic material out of that nucleocapsid in order for this virus to be able to carry out its infection cycle. So the uncoating process is unique to animal viruses because bacteriophages leave their capsid on the outside of the cell. Their capsid never enters into the cell. It stays outside. So animal viruses, the entire capsid enters into the cell. Also, penetration of the animal virus occurs in two different ways. We can either have what's called receptor-mediated endocytosis, which is what you're seeing here, or we can have fusion. And fusion only occurs in enveloped viruses. And what happens, oops, I moved you guys. What happens in enveloped viruses is these viral spikes will bind to receptors, and then the envelope, which is a phospholipid bilayer, will fuse with the cell membrane and drop the capsid into the cytoplasm. Whereas in receptor-mediated endocytosis, you should remember from your anatomy classes, is where multiple receptors bind, and then the animal cell will phagocytize or endocytize the entire virus and take it in. Okay, so we have two different forms of penetration. Then we have biosynthesis. So here, letter C here, we have biosynthesis occurring. We're making all of these envelope proteins. We're making copies of the genetic material. We're creating new capsids. And then we have assembly and maturation. So in assembly and maturation, here is our nucleocapsid with a copy of the virus genetic material in it. Down here, these viral spikes, remember, these viral spikes are going to be embedded in the envelope. They're embedded in the envelope, and the envelope comes from the cell membrane. So the viral spikes, if you notice here, get embedded into the host cell membrane, so that when the capsid buds out of the cell, it takes the host phospholipid bilayer, the host membrane with it, and its new envelope is host cell membrane with virus spikes embedded in it over the viral capsid and genetic material. So it's using your cell membrane for its envelope. If the virus is an unenveloped virus, meaning there is no envelope, it's what we refer to as a naked virus, then usually it is just exositized and there's no membrane around it, there's no envelope. Or, like in the case of herpes viruses, the cells lyse open and the unenveloped viruses are released into the environment. Okay, class time is up. It is over. We're done at 1:45.